Welcome to Biology, the scientific study of life. More about major groups of bacteria. This represents, notice that protobacteria. This represents alpha probacteria, and this one represents beta probacteria, and gamma protobacteria, delta protobacteria, and epsilon protobacteria. Also notice that gram-positive bacteria and cyanobacteria. These are the major groups of bacteria. More about extremophiles, meaning lovers of extreme conditions, from the Greek philos, lover. Extremophiles include extreme thermophiles, extreme halophiles, and methanogens. Extreme thermophiles, from the Greek thermo, hot, derive is very hot in environments. For example, Archaea in the genus Sulfobus live in sulfur-rich volcanic springs at temperature up to 20 Celsius degree. Biorolaps Phomera, an extreme thermophile found around deep sea hydrothermal vents on the mid-Atlantic ridge, can survive at temperatures as high as 113 Celsius degree. Another extreme thermophile, Bryococcus furces, is used in biotechnology as a source of DNA polymers for the polymers chain reaction PCR technique. Extreme halophiles, from the Greek halo, salt, live in highly silane environments such as the Great Salt Lake and the Dead Sea. Some species merely tolerate salinity, while others require an environment that is several times saltier than seawater. Colonies of certain extreme halophiles, from the purple red scum that owes its color to Bacteriodapsin, a photosynthetic pigment very similar to the visual pigments in the vertebrate retina. Methanogens are named for the unique way they obtain energy. They use CO2 to oxidize H2, releasing methane as a waste product. Among the strictest of anaerobes, methanogens are poisoned by O2. Some species live in swamps and marshes, where other microorganisms have consumed all the O2. The marsh gas found in such environments is the methane produced by these archaea. Other species of methanogens inhabit the anaerobic environments within the guts of cattle, termites, and other herbivores, playing an essential role in the nutrition of these animals. Methanogens are also important decomposers in sewage treatment facilities. All non-extreme halophiles and methanogens are members of a clade called Euraria Eukaryota, from the Greek Eurias, broad, a reference to the habitat range of these prokaryotes. Eukaryotes include some extreme thermophiles, though most thermophile species belong a second clade. Crina eukaryotes means spring. Crine means spring, as in hydrothermal springs. Genetic prospecting has revealed that both Eukarya Archaea and Crina Archaea also include many species of Archaea that are not extreme phylos. These species exist in habitat ranging from farm soils to lake to sediments to the surface waters of the open ocean. Now, new findings continue to update the picture of archaeal phylogeny. In 1996, researchers sampling a hot spring in Yellowstone National Park discovered archaea that do not appear to belong to either Eukarya or Ura Eukarya or Crina Eukaryota. They placed these Eukarya in a nucleate Korara Eukarya from the Greek Quran, young man. The oldest lineage in the domain Archaea, Korara Archaea, may offer clues to the early evolution of life on Earth. In 22, researchers exploring hydrothermal vents of the coast of Iceland discovered archaeal cells only 0.4 nanometer in diameter attached to the much larger Crina Archaea. The genome of the tiny archaean is one of the smallest known of any organisms, containing only 500,000 space. Analysis of the genome indicates that this prokaryote belongs to a fourth archaeal clade, now called Nanoarchaea from the Green Nanos dwarf. Within a years after this clade was named, 3 other DNA sequences from Nonara Archaea species were isolated, from one from Yellowstone's hot springs, one from hot springs in Siberia, and one from hydrothermal vents in the Pacific. As prospecting continues, it seems likely that the trio in this figure will undergo further change in years to come. Prokaryotes play crucial roles in the biosphere. Chemical, or if humans were to disappear from the planet tomorrow, life on Earth would go on for most other species, but prokaryotes are so important to the biosphere that if they were to disappear, the prospects for any other life surviving would be them. Chemical recycling: the atoms that make up the organic molecules in all living things were at one time part of inorganic compounds in the soil, air, and water. Sooner or later, that is where those atoms will return. Ecosystems depend on the continual recycling of chemical elements between the living and non-living components of the environment, and prokaryotes play a major role in this process. For example, chemoheterotrophic prokaryotes function as decomposers, breaking down cross-species dead vegetation and waste products, thereby unlocking supplies of carbon, nitrogen, and other elements. Prokaryotes also convert inorganic compounds into forms that can be taken up by other organisms. Autotrophic prokaryotes, for example, use CO2 to make organic compounds, which are often passed up through food chains. Cyanobacteria produce atmospheric O2, and some species also fix nitrogen into a form that other organisms can use to make proteins. Symbiotic relationships: just as a certain species of prokaryotes have beneficial associated with other prokaryotes metabolic cooperation, some prokaryotes form similarly intimate relationships with eukaryotes, and ecological relationships between organisms and different species that are in direct contact is called symbiosis, from a Greek word meaning living together. If one of the symbiotic organisms is much larger than the other, the larger is known as the host and the smaller is known as the symbiote. Symbiotic relationships are categorized as either mutualism and commensalism, and parasitism in mutualism. Both symbiotic organisms benefit, and in commensalism, one organism benefits while neither harming nor helping the other in any significant way. Commensalism is rare in nature, as further discussed in chapter 53. In parasitism, when organisms called a parasite benefit at the expense of the host. The well-being of many eukaryotes, yourself included, depends on mutualism. Depends on mutualistic prokaryotes. For example, human intestines are home to an estimated 500 to 1,000 species of bacteria. Their cells outnumber all human cells in the body by as much as 10 times. Many of these species are mutualists, digesting food that our own intestine cannot break down. In 23 scientists at Washington University at St. Louis published the first complete genome of one of these gut mutualists bacterioids, the Titomicron. The genome includes a large area of genes involved in synthesizing carbohydrates, vitamins, and other nutrients needed by humans. Signals from the bacterium activate human genes that build the network of intestinal blood vessels necessary to absorb food. Other signals induce human cells to produce antimicrobial compounds, to which B, that Titomicron is not susceptible, keeping other competing bacteria out of the intestines benefits but B, that Titomicron, as well as its human host.